We've learned how Toto and his team are able to take both venom and genetic data from live cone snails in the Philippines, and by various means arrive at a short list of promising chemical compounds called peptides that they think have medical potential. We saw how Toto and his team collect cone snails in the Philippines and process them to seek out the peptides in their venom with medical potential. They now have one or more peptides that are potentially useful as medicinal leads. In this next stage, we'll see how the various neuroscience experts in Toto's lab use their specialist expertise to analyze the peptides and find out just how they perform. The holy grail of cone snail research at the moment is a non-opioid painkiller that could help solve the opioid crisis. To understand how peptides can counteract pain, we need to understand pain. You feel pain when an electrical signal travels from a pain receptor in your body to your brain via a network of nerve cells. What all of this means is that scientists need to find out exactly how peptides affect the nervous system. This means that they won't need to use animals. Instead, they can test the peptides directly on cells. This is Peter. Peter is working with the peptide RG1A, a promising pain drug candidate.
So it's a peptide called RG1A that's specific to the alpha-9, alpha-10 nicotinic acetylcholine receptor, and we think it holds a lot of promise as a non-opioid pain therapeutic.
He's working to find out what makes it effective in blocking cell signaling. Many cells use electrical current to affect their behavior, which he can measure via electrophysiology. He inserts electrical probes into individual cells to measure electrical activity. Normally, the cell will transmit electrical signals when activated, giving Peter a graphical readout. He repeats the process, only this time adding a sample of RG1A to the dish where the cell is being held. He measures electrical current once again, but the reading shows there's no activation, which tells Peter that RG1A has blocked the transmission of a signal. In this case, Peter discovers that the RG1A can block signals that can lead to further nerve damage. Based on previous work by his colleagues, Peter knows that the natural peptide works. Peter is working to modify the molecule in the hopes of altering its properties. A version that lasts longer, for instance, wouldn't have to be injected as often.
The beauty of being able to synthesize these molecules in the lab is, since they're short peptides, it becomes very manageable to swap out these amino acids for different ones or even insert non-natural variations. So we can potentially improve upon what we find in the venoms. We can tailor it to better suit our needs as humans in need of therapeutics.
And he can accomplish all this in a timely manner because electrophysiology shows him exactly how each peptide affects the nerve cells. There's just one problem. Have you ever seen a nerve cell? They are tiny, usually too small to see without a microscope. So you can imagine why attaching an electrical probe to one would be difficult. Luckily, they can solve this problem with a bit of borrowed knowledge from other biologists. As early as the 1950s, embryologists had come up with a method of transplanting genetic information, RNA, into unfertilized frog eggs, which are 1,000 times bigger than typical cells. It turns out that the same technique could be used to transplant human RNA into a frog egg.
When Michael was doing alpha-9, alpha-10 nicotinic receptor, so I knew that he'd want a system, and so that would be like taking cells and putting in alpha-9, alpha-10 DNAs in there and see if I could express them. And if I could express them, then you could screen toxins, you could do all different kinds of things. The only place that you can do it reproducibly is in oocytes. Frogs is in the frog oocyte.
We take these frog eggs that are unfertilized cells, so it's basically a blank cell, and then we can then take the RNA of these different receptors and channels, and we can then microinject them into the eggs. And then the cell's machinery will then take that RNA and start expressing that receptor and then push it into the cell surface of the membrane of the cell. And then we then have a cell that's going to express our specific subtype that we're interested in.
Since frog eggs are 1,000 times bigger than human cells, this leaves you with a very big cell that you can use to measure signaling, which is much easier to perform electrophysiology on.
And we can then apply our conotoxins to check whether certain conotoxins will then block that receptor or if it will not block it.
This ingenious neuroscience technique lets undergraduates with less specialist skills get involved in the electrophysiology action, meaning more data can be gathered about the conopeptides in a shorter time frame. That's not the only way to study how conopeptides affect nerve cells. Here's Leavitt.
My name's Leavitt. I work for Toto as a laboratory specialist on the Constellation Pharmacology project, and I'm currently pursuing a master's degree in computer and data science. So our assay is getting to the point where it's almost as fun as a video game. The other type of technology we use is we have to apply these natural products to our cells. So to do this, we have some automatic fluidics. These are robots that control the application of fluids onto our cells.
He's also studying conopeptides, but he wants to study several types of nerve cell simultaneously.
The sensory organ of the body sends projections out to your fingertips, to your mouth, to your face, and it helps you sense pain, heat, cold, and touch. So we take this organ, it's also known as the dorsal root ganglia, and we break this organ up very gently. So what we do is we take this intact tissue, we break it up so that all of our cells are separate from each other, and then we look at these cells as we apply natural products to these neurons.
Different types of nerve cell respond to different stimulus. One might respond to heat, another to pain. And the conopeptides might affect different types in different ways. He uses a new process developed here at the University of Utah called Constellation Pharmacology.
So Constellation Pharmacology is revolutionary. And the reason why it's revolutionary is because previous assays have looked at homogeneous cultures. These are cultures where all the cells are the same. So the amount of information that you're getting from these is very limited. Now when you look at a tissue that comes from an animal, which is very heterogeneous, this means very different from each other. This, in a sense, gives you 15 different experiments to do at once.
The advantage of using constellation pharmacology over testing individual nerve cells is that several types of nerve cell can be studied at the same time, again allowing a greater amount of data on the effects of conopeptides on the nervous system to be gathered in a shorter amount of time. Now, I've mentioned the conopeptide RG1A a couple of times already. Let's hear from the experts about what made this conopeptide a strong candidate for a pain drug.
And so what we've done is we've used the cone snail peptide as a model, as a template, and gone in and introduced new changes into the molecule that allow it to work both in animals and in humans. It's important it work in the animals so that we're able to test it, show that it's effective, but of course, ultimately what we care about the most is that it works in the humans. So we've made those changes. It's now in the process of going through additional testing to see how long it lasts in the body, how often you would need to dose it, do we need to make additional changes in order for it to be an effective medication.
We don't know exactly how RG1A works, but we can see the downstream effects of the drug. So people who have this chronic pain typically will have some sorts of nerve damage, and we can see that we can relieve this like inflammation and damage with RG1A.
We think this is acting in a non-traditional way. We think instead of actually acting directly on the nerve, instead it's acting on immune cells. And those immune cells are involved in the repair of the nerves. And the receptors for the peptide are actually on the immune cells, and it signals them in some way to repair in a more effective and efficient way.
So, they finally did it. We've seen how the team found the kana peptides with medical potential and tested them all to see how they affect nerve cells. Now Todo's team know exactly which kana peptides can move forward into human clinical trials, which could clear them for use as a medicinal product. Although we don't have time to go into it in detail now, kana peptides are also being researched for other medical applications, such as cardioprotection and the treatment of diabetes and epilepsy. Completely different types of tests are used in these cases. One interesting example relates to diabetes research. Conus geographus, the world's deadliest snail, wages chemical warfare on the fish around it. When a fish is nearby, it releases a kana peptide that acts like insulin into the surrounding waters, dropping the fish's blood sugar levels and making them sluggish.
A few years ago now discovered that some of these cone snails, fish hunting cone snails, were making insulin. So I've done a lot of work on those fish hunting species that use insulin in their venoms. So insulin is a compound that is made in our own human bodies. So we want to find other venom compounds that look like things that we make in our own human bodies.
For the geographer cone, it's a way to make their meal slow enough to catch. For scientists, it's a potential lead for research into diabetes. Unlike the neuroscience processes we've looked at earlier in this episode, this test needed a fish research lab. Scientists confirmed that it acted like insulin on the fish by adding it to the water and observing a drop in the fish's activity levels.
We just looked at the compounds that were made by the cone snail. So we just looked at everything that was there and then noticed that one of those compounds looked like fish insulin. And so then, because it looked like fish insulin, we decided to test it at the insulin receptor and in the context of diabetes, which is nothing we would have done if we had used the traditional way.
So now you've seen exactly how cone snails get used in medical research, from collection in the Philippines to complex and innovative testing in the lab. It's exciting to know that several kana peptides are already in the late stages of development towards promising new medicinal products that could help combat the opioid crisis. And it's fascinating how expertise and research techniques have been built up over time, the research now involving experts from many different fields of science, all coming together to make breakthroughs beyond mere drug discovery.
We think that the peptides are pointing us in a direction that could be helpful for a variety of nervous system disorders if we could understand that underlying biology.
I think that's what interests me most with venom compounds, that once you have a venom compound, you may potentially have a drug lead, but you can also elucidate new biology. So you can find out new things, and you can only do that because you now have the venom compound that you can use as a tool. And so the two things always go hand in hand. The drug discovery part goes hand in hand with potentially identifying new discovery, new biology in humans.
In the next episode, we'll look at the stories of some of those key people in the Killer Cures story and see how they transform their childhood interests into groundbreaking science careers. I'm Max Taylor, signing off from another episode of Killer Cures. I hope you've enjoyed watching it, and I'll see you next time for more Killer Cures.